Hi folks, this is our last lab work. For this one, the virtual lab is at labexchange.org. Again, I have the link in there for you, and what you have to submit to me is a screenshot of this screen. Here, my actual results are blank because I haven't done the lab work, but your actual results will not be blank. Your actual results will look something like this. It will not look exactly like this because there will be something funny going on over here, actually. So that's what I'll be looking for. All the bands and the words actual results right there. That's what I'll be looking for. Now, just like last time, I would like for you to put your email address in one of these tabs when you take the screenshot. So there it is. There's Glenn right there. I take this screenshot, and then the teacher knows that it's Glenn who did this lab work because Glenn's tab is right there. So that's how to submit this lab work. And let me give you a few hints about the lab work itself. So let's see. Let me restart this whole thing. Okay, so a couple of hints about this lab work. Everything here is being done just the way you saw in the pre-lab videos, except for two things. And those two things are, there is a control antibody here, but there wasn't a control antibody in the pre-lab video. Now, the purpose of the control antibody is to verify that the Western blot technique is working. So we use the control antibody just like we would use the primary antibody. The secondary antibody, it will stick to both the control antibody and the primary antibody. All right? The control antibody is specific for a protein that is in all the cells. In this particular experiment, we're imaging the gel using the Western blot from an experiment where we extracted the proteins from wild-type cells, from edited cells, from some other edited cells, and I think that's it. The edits are supposed to remove a protein. The edits are supposed to remove a protein that is useful for the HIV virus to infect. So presumably, if you don't have the protein, you cannot be infected by HIV. So the edits are done by CRISPR-Cas9, which is a new technique that recently won a Nobel Prize, but it is not important for the lab portion of our course. It's just important that you know that it edits genes. So the edited cells are supposed to have no... The protein in question here is called CCR5. So the edited cells are supposed to have no CCR5. That means HIV won't stick to those cells. That's good. But the wild type, unedited cell is supposed to have CCR5. Right? So the control antibody is specific not to CCR5 because CCR5 is the purpose of this experiment. The control antibody is specific to a protein that is in every cell. In this case, this control antibody is specific to tubulin, which is I think it's like a cytoskeleton structural protein. But it's a protein that we're not touching with these edits. So all the cells should have this protein. So the reason why, one more time, the reason why we're using this control antibody is to verify that our process of Western blotting works. The reason why you have to do that is because suppose you do a Western blot experiment and you show the other scientists, when you publish this article, you show the other scientists your work and they're like, yeah, I see that you're trying to show us that your edits remove this protein, but actually, your work just shows us that your Western blot technique doesn't work because I don't even believe that you actually edited anything. So in order to prove that the technique itself worked, we used a control antibody to show that a protein that has not been edited is present and the Western blot does show it. So control antibody is just like primary antibody, except it is specific to some very common protein that we're not touching. Okay, so that's a nice hint for you here. Another hint for you here is this. When you use the Western blot transfer apparatus, you're going to plug this in just like the SDS Page electrophoresis box, and when you plug this in, you have to see that the brass plugs are disappearing. All right. So let me show you what not to do. See that is good. Let me show you what's bad. See that is bad. It says here that there is a cable connected, but that's not. I tried this several times, and it won't let you turn this on if you could see the brass plug. The brass plug has to be disappeared. Has to have disappeared. Yes, like that. You see that brass plug had disappeared? Yeah. You got to bury that brass plug into the barrel there, like that. All right? Okay. And then I have another hint for you. When you try to turn this on, you're going to have to change the voltage like that. And then you're going to have to go to 150. You're going to have to change the time. Now, this timer here is very hard to see because it's small. We're looking at these numbers right there. That's what you have to be changing, all right? Then you will do the start. And just like last time, the time goes down real fast, okay? All right. Now, you can express yourself as you do this lab work. I mean, expressing yourself, just sort of optionally. It's just something that you do to make the lab work more interesting for you. So there are some things in here by which you could express yourself, and let me show you what those are. For example, you can, when you obtain some stuff, you can close the cap. If you're a careful lab person, after you finish with a reagent, you close the cap. If you feel like you're a messy kind of lab person, then you don't close the cap. You leave it open. When you're done with liquid waste jar, you close the cap. If you feel like you're a messy guy or a messy girl, you don't close the cap. You know, so you can do a little bit of self-expression here. And some stuff, some of these lab tools, they could be wherever you want. You know, it won't affect your results. But you got to show me the actual results, and those bands need to be correct, right? And you got to show me your little email address tab here, okay? So this is quite tedious, but not half as tedious as doing the actual lab work is. Although doing the actual lab work is more fun. Okay, bye.